So we are joined tonight by Rachael Tarlinton. Rachael is an Associate Professor of Veterinary Cellular Microbiology and a European diplomat in veterinary microbiology. Her research expertise is in animal virology, particularly retroviruses, but covers a broad range of virology and genetic methods in domestic and exotic species, with a current project on retroviruses in koalas, cats and sheep, as well as SARS-CoV-2 monitoring in wildlife. I hope I pronounced that right. She graduated with a Bachelor in Veterinary Science from Sydney University in 1999 and worked in mixed practice in Australia and the UK. She then did a PhD on retroviruses of koalas at the University of Queensland, and then spent time after this working as a postdoctoral researcher for the Queensland Department of Primary Industries at the George August University in Germany. She joined the School of Veterinary Medicine and Science at Nottingham in 2007, where she teaches undergraduate and clinical phase veterinary medicine students and runs a research programme in animal infectious diseases. Rachael's talk tonight will summarise what is known about the SARS-CoV-2 in animals, including work from a current project on UK wildlife, and she will discuss what is likely to happen going forward. I'm now going to pass over to Rachael.
Excellent. That all sounds very impressive read out like that. It was rather a lot of work at the time. So as Joe said, I'm going to talk to you about coronaviruses in wildlife, and particularly SARS coronavirus monitoring we've been doing in UK wildlife. So just to start off with, I'm assuming most people in the room are not virologists. So this is just a little bit of an introduction to coronaviruses and SARS coronavirus. So just bear with me because some of these bits are important for explaining some of the stuff we've been doing later on. So coronaviruses are enveloped RNA viruses. What this means is they're really fragile in the environment. They have a lipid membrane around them that they take from the cell that they need to replicate, and that's really easily destroyed. So they're really easily destroyed by soap and drying out and things like that. What it means practically is they don't last more than a few minutes, hours at max outside of their host, so they don't really spread on things or fomites. For an RNA virus, SARS is actually really big. It's got quite a lot of genes. I'm only going to talk about a few of them that are important for what we've been doing with diagnostic testing. So this graphic over here was generated by my colleagues at the University of Glasgow, by Ed Hutchison, in collaboration with a visual artist from electron microscopy pictures. And these are actually pretty accurate for what it looks like under an electron microscope without the colors. Those are artistic license. So it looks like this kind of fluffy pom-pom thing. And then some of the important genes that we need to talk about are the S gene, or the spike gene, which is the one in the vaccines and the ones people talk about a lot. And it's the one that sticks through the envelope, and it's the one that actually binds to the host cell. So it's really important for actually being able to get in and out of host cells. And it's also really important because it's sticking out of the outside of the virus. So it's the bit your immune system sees, and it's the bit where there's the most pressure on the virus to change or mutate to get around the immune system recognizing it. So it tends to change quite rapidly with viruses like this because it's trying to get around the immune system. Another important gene is the envelope gene, which are these little, oh no, that's membrane protein. So envelope gene is here in the envelope as well. It's part of the mechanisms for actually packaging up all the RNA. So this cotton wool stuff in the middle is the actual genetic material. It doesn't vary as much as the spike protein because it doesn't stick out as much. So, and it's quite a small, very, very small gene. So we use that for different things as well. And the other one we're going to talk about tonight is the very inelegantly named RDRP gene, which is the RNA-dependent RNA polymerase. And basically, it's the enzyme the virus has that it actually needs to make new copies of itself. So it's the bit it absolutely needs to replicate. And it's hidden and packaged in the middle with all the genetic material. So it doesn't vary much because it's essential for the virus and it's very hidden from the immune system. So it's generally the least variable bit of the virus. So that's, you'll be relieved to hear that's it for virology bits, but it's a bit important for some of the other stuff. So there are a lot of coronaviruses, and these are just the ones we know about. This is from a paper from a few years ago, and it's only a selection of the ones we know about now. So every species we go and look at, we find coronaviruses if we look hard enough, pretty much. So there's a huge number of them, and they infect a lot of different species. In mammals, they're generally thought of as causing gastrointestinal or respiratory disease. Some species, like humans, have up to 4 or 5 of them, things that we've studied well. And this picture over here is a phylogenetic tree, which is a graphical way of graphing out how closely related viruses are to each other. It's done with a big computer program. But there's basically 4 big groups of coronaviruses. So we have our Delta and Gamma coronaviruses. These are primarily viruses of birds. We only know of a couple of examples of those in mammals, and that seems to be quite rare. So they're mostly viruses of birds, and we're not really going to talk about them very much tonight because there's no examples in people. The ones we're more concerned about in mammals and people are the alpha and beta coronaviruses. And you can see there's just a huge range of different viruses there. So, a bit more about coronaviruses. Most of them are mammalian coronaviruses, belong to the alpha and beta coronaviruses groups. Most species that we look at have them, as far as we know. Lots of them have more than one. Humans have at least 5, both alpha and beta. Dogs have both alpha and beta coronaviruses. Probably other species have lots more, but we actually haven't done a lot of testing or looking outside of rodents, bats, and domestic species. So when you actually start looking, there's huge gaps in our information base for what's out there. The ones we really worry about, or the ones that you hear in the news, are the ones that have spilt over into humans recently. And most of these are thought to probably have originated in bats. So we've got MERS coronavirus. So it's actually a camel virus that people contract from camels, primarily in the Middle East. We see sporadic cases of that every year. We had SARS-1 in 2006, which we think went from bats to civets, which are a type of small carnivore, and then into people in southern China and Hong Kong. And then, of course, we had SARS-2, which we've all suffered from for the last couple of years. We may never know exactly what the intermediate species was. We think it probably, like SARS-1, went from bats into something else in that market in Wuhan, but we will never know what it was because they cleared it quite quickly, and then on into humans. So obviously there's been a lot of attention because of the scale of the damage of particularly the SARS-2 outbreak to everybody and everything on wildlife coronaviruses and how these things get about and where they come from. So bats get blamed for a lot of viruses, perhaps a bit unfairly, because the basic issue is there are a lot of species of bats and a lot of species of coronaviruses. So we think that we have about 4,000 species of bats worldwide. So when we talk about bats and viruses, we're actually only talking about a tiny number of the ones that we know about, but there's a huge number of bat species and they have a huge number of viruses. So this map over here is not mine, it's from another paper. And this is just the horseshoe bats that we know. So this is the known host ranges of the different species of horseshoe bats. And you can see just how big a diversity of different species and the range they cover just in the horseshoe bats. If you start looking at fruit bats, they can travel 400 or 500 kilometers a week as individuals. So you start seeing the scale of the problem. The ones we worry about in terms of spillover into other species in humans are primarily beta coronaviruses. So both SARS and MERS are beta coronaviruses. And just a taxonomy thing, so beta coronaviruses are further split into sarbecoviruses, merbecoviruses, norbecoviruses, mbecoviruses, and hibecoviruses. So primarily we're going to talk about the sarbecoviruses tonight. And sarbecoviruses, the SARS-like ones, are pretty much, we're pretty confident they're pretty much horseshoe bat viruses. So all the natural host reservoirs of sarbecoviruses are all horseshoe bats. We have sarbecoviruses in our European species of horseshoe bats. The ones we have are not thought to have the particular genetic features in the spike gene, so the furin cleavage site, if you follow that sort of thing, that convey infectivity to people. So we don't think the ones we have in Europe are infectious for people, but we haven't actually tested that, and to be frank, we're not going to. So SARS itself, since it emerged, the scale of the human outbreak has been so overwhelming and so massive, it's then gone on from humans into lots and lots of other species. So we've had field reports of this huge list of species, some more exotic than others. The ones we've seen a lot of reports in include carnivores, particularly felids, so a huge number of different zoofelids, domestic cats, we'll talk about them a bit later, and other carnivores like dogs, ferrets, mink, hyenas. We've also seen an outbreak in hamsters, which we'll talk a bit more about later, zoo cases in great apes, and we'll talk a bit more about the white-tailed deer in the US a bit later. Some of these field outbreaks have been quite large. We know from lab work that quite a range of species are potentially susceptible, so are able to be infected in a lab, and there's quite a list there. The list of species we have infected or can infect in the lab is not exactly the same as the list we've seen field cases in, and we make a lot of predictions as virologists about which species should be susceptible, and it doesn't always pan out in terms of which species we actually see in the field. There is a question about house mice being infected, and that was a really big question at the start. It would appear that the earlier strains of SARS-2, so the Wuhan strain, can't infect lab mice or house mice, but some of the more recent strains, so Omicron, Delta, Beta, can infect mice in the lab, but we've not seen any field evidence of this. Some people have been looking in mice and rats, and we've seen the same thing in primates, while we've seen it in zoo and lab, the common lab models. We've been doing some work with colleagues in India, and we've not detected it at all in their urban macaques there. So there's quite a difference between what happens in the lab versus what's actually happening out in the field. So some of the really important examples in terms of actual disease risk and epidemiology: cats. So this is one of my cats. They're readily infected in lab experiments, and they readily spread it to each other. And we've had lots of reports of domestic cats contracting it from their owners. And there was a very recent report, came out probably about a month or two ago, that was a confirmed case of a cat to human transmission. That was a veterinarian in Thailand that got sneezed on by a cat from an infected household, and it's pretty tight, that one, as to that's definitely how she got it. Doesn't seem to be terribly common for that to happen, but it's certainly possible, and we have at least one documented case of it. Cats do pick it up from their owners reasonably often. So it's Margaret Hosey at Glasgow that's been doing all the cat monitoring. And the last time I talked to her, she said about 5% of all the cats in the UK are seropositive for SARS-CoV-2. So they've had the infection in the past and recovered from it, which is actually quite a high number. it's probably not spreading from cat to cat as far as we can tell, probably mostly because cats are quite solitary and they don't spend a lot of time with each other. It could be quite different in a big cattery somewhere, but we've not seen reports of that. So cats definitely catch it. They can spread it to people, but that's quite rare. They're probably not spreading it to each other as far as we can tell. It's a bit of a debating point as to whether it causes disease in cats. Most of them probably, like with people, most of them probably don't recover quite quickly, but there have been some reports of cardiac problems and respiratory problems from some vets, but that's still quite an open debating point. We do know that the whole Felidae range seems to be quite susceptible to these, because we've had lots and lots of reports in different zoo and wild felids of sporadic cases of it. So cats seem to be quite susceptible to this type of virus. Mink. Some of you will be aware of the enormous panic that occurred with the outbreaks in the farmed mink in the Netherlands, and the investigation of that was initially triggered by a report of a respiratory disease outbreak in those mink farms, and our Dutch colleagues are very, very good and we got onto this extremely quickly. It spread like absolute wildfire through these farms. In one case that the Dutch guys published, 100% of the animals tested positive within the space of a couple of weeks. Most of them got over it, recovered, but the rate at which it spread was really, really fast. And there were documented cases of transmission back to some of the people who work in the mink farms, which prompted Denmark and the Netherlands and a number of other countries to cull out any affected mink farm. In some cases, like in the Netherlands, they were phasing out fur farming anyway. We stopped it altogether in the UK in 2006, so we didn't see this problem. So they, in essence, brought forward the closure of that entire industry. We have seen outbreaks in most places that farm mink. Some countries are better at reporting it than others. China and Russia, understandably, have been very poor at reporting it, but we know they've had cases and had problems. So it's not just something that's confined to one place. Interestingly, we've had no reports in wild mink. We've looked, the USDA have looked, the Dutch have looked, and the Danish have looked very intensively in and around those affected mink farms. There's one probably farm escape mink picked up positive in the US. That's all we've seen. So it isn't spreading at all as far as we can see in the wild mink, which is probably fortunate. The reasons for the difference between the way the virus is behaving in the farms versus the wild is probably really to do with the species behavior, like with the cats. Feral or wild mink are a solitary species. They don't have a lot of contact with each other. And that's probably what's going on there. So we've got a big difference between what's happening in our captive animals versus our wild animals. Just a note about ferrets, they are just as susceptible as mink and cats, but they don't tend to be kept at the same scale or the same sorts of numbers. So we've not really seen a lot of reports of cases in ferrets. They're just as susceptible, they're the most common lab model, but there just aren't as many of them as there are of these other species. So we've not seen a lot of reports in those. So deer. So this is my colleague Suresh Kuchipudi, who used to work at Nottingham and now works in the US. This is a publicity photo from one of the newspapers. He's done a lot of the work on this, but it kind of neatly demonstrates the scale of the problem. So we've had a very large outbreak of SARS-2 in white-tailed deer in the USA, and I haven't checked recently because they haven't published their most recent figures, but as of about August, something like 40% of animals in some places were seropositive, so they'd had it. So that's a very large number of animals. And this not just in one place in the US, it's been reported in Canada, in 5 or 6 different states by 5 or 6 different research groups working independently. So it's definitely very real, and they pick up virus and can grow virus and serology, and it's very real. We know from experimental work that the white-tailed deer spread it to each other quite readily experimentally. They are a really common species in the US, and they're at very high densities in urban and peri-urban areas. So this was taken in a city park. Lots of people feed deer in their backyards, even in big cities. And there's, I've forgotten how many, something like 20 or 40 million animals across the east coast of the US in quite high densities. They're also hunting, whatever you think of it, is a popular activity in the US. And populations in a lot of rural areas are managed to maximise the numbers for hunting. Many people at this time of year will still go out and shoot a deer to put in the freezer for their winter food. It is a very popular activity. And we don't know how it got from the people to the deer. We know, looking at the virus phylogenetics, that it happened lots of different times in lots of different places. We also know that it's definitely spreading between the deer now, and we've seen quite a divergence between the strains we see in people and the strains we see in deer. And the risk or the problem there is, if those strains then come back into people, they're going to be quite different to the ones in people, and we might see a big surge in cases in people because they won't be as immune to the deer variants. So far it hasn't happened, but obviously it's a big worry, and colleagues in the US are monitoring that very intensively. Interestingly, in Europe, none of our deer are positive, so we haven't detected it at all. And there have been some quite large studies looking at that. So the UK Health and Safety Authority, who used to be Public Health England, were monitoring deer for tick-borne diseases and kept going monitoring for SARS. So they tested 1,500 animals, and not a single one showed evidence of infection. Colleagues in Germany similarly have been testing hunted deer there and haven't seen a thing. I know colleagues in Scandinavia were looking at their reindeer, and I haven't seen any reports at all which they would have reported it if they had found it. So we're not seeing anything like this in Europe, probably, thank goodness. And nobody really understands why we're seeing such a difference between the situation they've got in the US versus the situation we've got here, because our deer, in theory, should be able to be infected, but we're not seeing it. We do have a different species range to the US. We don't have white-tailed deer. The UK has 6 species, a lot of which are not present. Things like the muntjac are not present in the rest of Europe at all. So maybe they're just inherently not susceptible. It may also just be to do with this population density issue, that our deer, by and large, are not as abundant as the white-tailed deer in the US. So that's deer. So hamsters and other rodents. So this was a bizarre one. So Syrian hamsters are actually the most commonly used lab model for human disease, and they quite readily spread it to each other. And it appears that cricketted rodents, so Syrian hamsters are cricketted rodents, are quite susceptible to this. So deer mice in the US, which are also a cricketted rodent species, also spread it to each other in the lab. But so far no one's picked it up in wild deer mice. The USDA are looking. I talked a bit earlier about house mice and rats, which are a completely different group of rodents. No one's detected it in them at the moment. There was some speculation about mouse sequences in sewage, but I think everyone's pretty happy now that those are more likely to be human strains that come from the gut, so they're different to the ones that come from the lungs. What did happen earlier this year, however, was they had an outbreak in Hong Kong, and it was a different strain than the human strain that was circulating at the time. And when they traced, China is still pursuing a zero COVID policy, and so they traced back these cases and traced them back to a pet shop. So all the cases with this strain originated in pet shops that kept hamsters. And when they tested all the animals in the pet shop, a number of the hamsters test positive. And it would appear that when we looked at it properly, it would appear that the hamsters had been imported from the Netherlands. I don't know why this is economically viable, but they breed hamsters in the Netherlands and then ship them to pet shops in Hong Kong and Singapore. And that's where most of the pet rodents in Hong Kong and Singapore arise from. And it had probably been circulating for quite some time in the breeding facility for the hamsters in the Netherlands. It had then been sent on a plane to Hong Kong and had then infected people. The Chinese government at this point ordered all of the hamsters in Hong Kong to be culled. Can't tell you how successful that was or not, but it certainly led to huge problems with people trying to hide their hamsters or refusing to hand them in, and weeping children having their pets being put down. And it was not a good way to handle this type of problem, particularly as the hamsters were generally fine and would have gotten over it and could be kept confined in their houses like the people. But I'm not the Chinese government, and that was their decision. So this is the work we've been doing in the UK. So we have been monitoring UK wildlife species. So a variety of different species. So the ones we were focusing on were the ones at the time we wrote the grant. This was before the deer outbreak that seemed to be likely to be the highest risk. So we were looking at mustelids, because at the time we knew about the mink outbreak. So in the UK, that's otters, badgers, weasels and stoats, feral mink, pine marten and polecats, foxes, horseshoe bats. We have 2 species of horseshoe bats in the UK, greater horseshoe and lesser horseshoe bats. We have 3 species of cricketed rodents, water voles, field voles and bank voles. And we set a target rate of 80 samples per species. We've done some previous work with rodent coronaviruses, indicating that if you have a virus that's circulating in the animals, an endemic virus, you're probably going to have about a 5% detection rate of the virus infection in those animals. So that's why we set the numbers the way they were. And we sampled from about 2021 onwards, though for some of the rarer mustelid species, we used historical samples because we couldn't get sample numbers. And these were, depending on the species, the samples came through via a variety of routes. So for the rodents, these were largely, we did some trapping ourselves, and various ecology groups that do regular rodent population monitoring provided us with fecal samples out of Longworth traps. So this is the PhD students and postdocs who are working on the project down on campus with some Longworth traps. So just trapped the rodents overnight, released them, and then took the feces out of the traps, because in pretty much all of these species, and SARS in people as well, you can detect it both in respiratory secretions and in feces. People don't like donating feces for samples, but it's the easiest way to collect samples from wild animals, and it's non-invasive. And all the guys doing the water vole monitoring programs sent us a huge number of samples from those. We sent them kit to store them in, and they posted them back to us. So we currently, which I think Malcolm Bennett is going to talk to you guys later in the year about, we currently are doing tuberculosis monitoring in badgers for DEFRA. So we have a large number of badgers coming through our post-mortem facility. So those animals were the ones that were used for coronavirus monitoring. Similarly, the Cardiff Otter Project has a long-running program for postmortems for otters that have been hit by car generally. So found dead otters all go to Cardiff, and we accessed samples through them. And bats similarly were collected by volunteer collectors monitoring bat roosts, sending us in fecal samples. So this is technical details. We extracted RNA, which is the virus nucleic acid, turned it into cDNA, and then applied 2 different PCRs for them. So that's a way of detecting the virus nucleic acid. So one of them was a PCR that for the RDRP gene that should pick up all coronaviruses, and we've used that before studying rodent alpha coronaviruses, and most groups that look at trying to find new coronaviruses in bats or things use that PCR, so we use that one too. The other one we used is a qPCR, which is a more sensitive method that's specific for sarbecoviruses. So it was developed by one of the German groups as a SARS diagnostic, but it will pick up all of the sarbecoviruses. And we ran some other PCRs for general things that should be in DNA or RNA, just to check that the samples were okay. So those tend to be called reference genes. So if you're interested in that sort of thing, it's cytochrome B or beta-actin. So these are the results, the important bits. So for most of the species, including, seeing as you're a badger group, you'll be interested to see our badgers were negative, with the exception of 3 stoat samples. And this was on the pan-coronavirus PCR, but not on the sarbecovirus PCR. So the stoats, all the stoats came from the Orkney Native Wildlife Project, which is actually a stoat eradication program that's led by the RSPB. So stoats are not native to the Orkneys. They were first reported there in 2010, and they have a really destructive effect on nesting seabirds. So they are trying to eradicate them. I'm not quite sure how successfully, but they are attempting to. And this needs updating. So we have now sent these viruses off for sequencing and retrieved a full genome of the coronavirus, which is quite a technical feat. My postdoc's done quite well there. And the virus is an alpha coronavirus. It's one that's very closely related to ferret and mink coronaviruses from the Minna coronavirus subgroup. So I'll just to explain, this is another phylogenetic tree. This is for this group of alpha or Minna coronaviruses. These group of viruses are actually quite interesting in and of themselves. So this type of alpha coronavirus is quite widespread in mustelids, the ones we've tested. This is what we found. And when they went back and checked what other coronaviruses all the mink and the mink farms that had SARS had, a huge number of them also had these too. These viruses cause disease. They are known to cause diarrhea in ferrets and mink. And the other somewhat concerning thing about this group of viruses is they're reasonably close related to dog and cat alpha coronaviruses. And these dog ones in particular are known to swap segments with related feline and porcine, so from pig viruses. And there've been those types of recombinants have also been reported in raccoon dogs and recently in human children, because there's increased monitoring of human respiratory disease. So we're picking more of this sort of stuff up. So this group of viruses are one that's kind of on a watch list for things that are quite prone to recombining and ending up in different species. So this pig virus, TGEV, caused a really devastating outbreak of diarrhea and disease in commercial pigs, and it caused a massive problem when it emerged. So our virus kind of sits in here. It's off a little bit from the mink coronaviruses, but it's definitely part of this group and seems to be quite stoat specific. So that's virus number 1 we found. Cricketted rodents, we found nothing at all, which is good. Even in the water bowls, and some of the water bowls we had, particularly, we had samples from the fossorial water bowls in Glasgow. So they're in public parks in Glasgow at the height of last January's outbreak. And we got samples back with things like found dead across from testing centre. So if we were going to see something, it would have been in these animals, and we got nothing at all. So that was quite reassuring. So that's rodents, so a big fat 0, which is a good thing. And clearly we're not seeing anything like what happened with the hamsters in Hong Kong. So it's another situation where we've got something really different going on in captive animals versus wild animals. So bats. So this gets quite interesting. So we did detect virus on the E-gene qPCR, the sarbecovirus PCR, only in the lesser horseshoe bats, not in the greater horseshoe bats. And the one we've picked up seems to be quite specific for lesser horseshoe bats and nothing in any of the small number of other species. There's actually quite a lot more samples we've done now because the project's nearly finished. So we probably have upwards of about 600 greater and lesser horseshoe bat samples. But this is a reasonably high prevalence rate we're picking up there. However, it's not SARS. Like I said, that E gene qPCR will pick up all sarbecoviruses. And a group at UAE and Public Health England published last year the sequence of a lesser horseshoe bat sarbecovirus. And we've now got our full genome sequencing back. And the one we've got is very, very similar. So it's basically the same virus as that one. And it appears to be a lesser horseshoe bat specific sarbecovirus. It's really interesting that we are only seeing it in the lesser horseshoe bats, not in the greater horseshoe bats, even though the two species share roost sites. And the virus really looks quite species specific. We actually don't know very much about sarbecoviruses in their bat hosts, how specific they are to bats, how they get into other species. And in this case, this particular virus looks to be quite species specific. The pipistrelle samples we tested were all for animals, from animals that were in care, where the carers had contracted COVID. So the IUCN bat guidelines recommended testing animals before being released in those circumstances, but there isn't actually a testing service that's accessible. So we stuck them through our research program. They were fortunately all negative, and those are really high risk animals. So it's looking like that species really isn't very susceptible. So these are maps of where we found them. So lesser horseshoe bats are not a common species in the UK. There's not terribly many of them. So we're talking about 17,000 individuals, and in the summer less than two. In the summer they use different roost sites between summer and winter. In the summer, less than 200 roost sites. In the winter, less than 300. So not all roosts were positive, but we found positive samples, which are these red spots here, across pretty much the whole range. So this is a map of the range which we would expect to find them in. These are the roost sites we had samples come in, and there's no obvious geographical pattern to which roosts were positive, which ones weren't. Some of the samples we had, we can't really work out individual versus roost because of the way the samples came in. But it's pretty widespread, and it'll be, for it to be this widespread in them, it must be endemic, and probably they will have a chronic gastrointestinal infection. We know that happens with cats. For us to be picking it up at this higher rate, it's almost certainly a chronic virus infection in these animals. We don't have a clue, however, what it does to them, whether it has any effect on them, whether it causes disease, whether it has any effect at all on them, whether cats that eat them pick it up or not. We have no idea. That's as much as we know about it at the moment. That it's there, it looks to be widespread. So so this is kind of where the project's at. We've now finished doing full genome sequencing for the viruses and are writing this up for formal publication at the moment. There's a whole pile of work we'd like to do to follow on, but obviously that's dependent on us obtaining further funding and me writing the grants in the first place. It's been a bit of a frantic couple of years getting all this happening. But there's a huge number of things we need to work out. Does it do anything to the animals? Does it spread? Does it vary from season to season? How's it spreading between the animals? And the other big question that we don't know is looking at those experiments with house mice, is as the virus changed in people, the range of species it was able to infect changed. So we don't know if the virus keeps changing in people, which it will. We're very sure of that, whether that will change the range of species it's potentially able to infect. So the situation we've got at the moment is what we know now. It may not stay that way. We might get further species or further things happening depending on how it changes in people going forward and what happens with that outbreak in the US. We just have no idea. So we're nearly coming to the end now. So I last gave this talk to a bat group, but the same principles apply. We can't really 100% stop people interacting with animals. We're not going to stop. We haven't even attempted to stop people interacting with their cats because it's just not going to happen. And similarly, trying to prevent people from interacting with deer in the US is just impossible to enforce. If you are involved with species monitoring programs, these are guidelines from the IUCN for prevention of spread from people to bats. And to be honest, they're pretty sensible. And these are the kind, I'm also a biosafety officer, so I deal with this sort of thing a lot. And these are the sorts of things they do for high risk, high value species like the gorillas in Rwanda for tourism. So wearing proper FP3 face masks would be very sensible to prevent you from infecting the animals, and basic things like changing your gloves, changing your boots, making sure your boots are washed, you have clean clothing and equipment between sites are really critical for preventing you infecting the animals and you dragging stuff from site to site. With other species and other diseases, we know this is a problem. This is how white-nosed fungus spreads between bat caves in the US. Free human testing is now at an end. So that's no longer an option for screening yourself before going into these sites. And these are kind of just general things for any disease, regardless of whether it's SARS or something else. So going forward, control options, we can't really stop it happening, particularly as it's not going away in people ever, and the pressure from the number of human cases is just too high. So this is just a constant risk going forward now. We've already talked about potential changes in species range. Can we vaccinate animals? It's very possible. They respond fine, works fine. Up till now, it's not really been considered ethical. Well, a lot of the human population hasn't been able to be vaccinated, but with the pressure coming off the human pandemic, that consideration is probably not quite so critical as it was. Zoetis have developed a vaccine. They developed it with an eye to a primary market in dogs, cats, and farmed mink. And to give Zoetis credit, they're not an evil pharma company, but they can't produce a vaccine at a loss because that's just not possible. To keep it in production, it costs millions to put these things through the regulatory process and maintain all the machinery and production quality control to produce it commercially. So if they don't have somewhere to sell it to, they can't make it. So they have developed one. It's not licensed in the UK or most of Europe. They have donated it for free for use in zoos for high risk or high value species, and have donated it for endangered species programs, in particular black-footed ferrets in the US. There is a Russian vaccine. No one knows how good it is. The human vaccine has serious concerns with fraudulent safety data and its registration data, so I'm not sure how much we'd trust a Russian-made animal vaccine, but they're primarily targeting their mink farms. With that is their primary concern. The other issue with vaccinating wild animals is it's just exceptionally difficult because you've got to catch them and jab them, and for a lot of species that's just not feasible or practical. So it's probably not your first line choice for your however many million white-tailed deer in the US. So that's it really. There's been a huge number of people working on this project, and it keeps getting bigger as it's gone on. A huge number of different there's no space to list all of the contributors and different wildlife groups that sent samples in. These, you know, the academics and the bigger projects that have contributed to us. And the project was funded by UKRI, by the BBSRC. We couldn't, this type of work is expensive to do. There's no two ways around it, and it can't be done without that large scale funding from government. So that's really it. So happy to answer any questions people have got, and I hope that was interesting and useful for you.
Extremely useful. Thank you so much, Rachel. I'm not from a scientific background, but I was really able to follow that really well. Lovely and visual. Thank you so much for that.
My pleasure.
Just going to hand over to Cheryl. We've got quite a few questions actually. Cheryl, how are we doing on questions?
Yeah, we've got quite a few. I've got firstly two that go together. What advice would you give people if they have COVID and are concerned that they might have passed this on to any pets? And then somebody asks on the back of that question, do domestic pets show symptoms?
Yep, so dogs are the easy one. So dogs can get infected, but they don't show symptoms, they don't pass it on to each other, they don't pass it on to people. So they're regarded as a dead-end host, and we're not too worried about dogs. Cats are reasonably readily infected. The vast bulk of cases we've seen in cats haven't shown any symptoms, and it's just been picked up on the testing Margaret's been doing on other things. So in terms of your pet's health, I wouldn't be too worried about them. Obviously, cats can spread it to each other and can spread it to people. So if you have a vulnerable person in the household, it would be a good idea to keep them separated from the pets. If you are really worried about your pets, the same types of precautions you would apply for preventing human-to-human transmission, so keeping them isolated and or wearing a mask would be appropriate. But there isn't any more risk to your pets than there are to you, and we've not seen huge spikes in reports of disease in cats or lots of, they can get it and they can spread it, but it doesn't appear to cause them a lot of problems for cats.
Brilliant. Right, so I'm going to go through these in order now. Has a search for virus in ticks or mites been done to inform transmission?
So not specifically for SARS, but there are quite a lot of monitoring programs for tick-borne diseases. So we know in the UK we've had a recent introduction of tick-borne encephalitis virus in deer in Lincolnshire, and it's quite widespread and spreading. So the reason the PHE were monitoring deer is they were also monitoring deer and ticks for tick-borne encephalitis virus. So there is a ongoing and quite large monitoring of ticks, and they tend to do that. You can either do what we did in PCR for a specific virus, or you can just sequence everything in the sample, which tends to be what they've done with the ticks, and that's ongoing, and no one's picked it up. So there's no indication at all that SARS is vector-borne, that it goes through insects. It's probably a good thing because vector-borne diseases are very, very hard to control. So there's no indication at all that it goes through ticks or mosquitoes, and there's lots and lots of monitoring of ticks and mosquitoes in general. We would have found it if it was an issue by now. And the APHA have recently got a 3-year grant to look at establishing a proper tick monitoring program, because my postdoc who's been working on this project is going down there to work on that project. So there is funding and plans to actually establish proper tick-borne disease monitoring and do this longer term. So that is at least underway, because we've known for quite a long time that we've got big gaps in our surveillance. So at least we're addressing that.
Another question here, of the badgers that were tested geographically, where were they from? How did their carcasses come to be available for testing? Is that road casualties or other sources?
So I would need to fish this data out from Malcolm. So the most of the animals that would have come in are from, so the project is the DEFRA Southern Edge TB survey. So the areas that are being focused on are the ones that are regarded as being at the edge of the endemic TB zone. So they're largely, some of them have come from down your way, from around London up through the Midlands. So it's a mix of different origin samples, because DEFRA collect the carcasses and send them to our post-mortem facility. Some of them are roadkill, some of them have come from culling sites. I don't know the exact mix, and I haven't been back and checked the numbers and origins. We've got over 600 badger samples in the data set, so there's a lot of them. We have in the past done, as Malcolm had run the Northern Edge survey previously. The vast bulk of them that come through are roadkill, but there are some from culling sites.
And a couple of our members are doing bat monitoring in Bridgewater with the with WWT, and recorded lesser horseshoe bats, and they're still doing this for 2022. And this is using the audio moth electronic devices, so there's no interaction, and they said they can share that data.
That would be cool. We're hoping to have a PhD student who's interested in continuing the bat project, and we're hoping to try and continue on with that, but it's still in the stages of being set up. So if there's groups that are keen to help out or participate, we'd certainly really like to be able to follow up seasonally and over a longer period of time what's happening in the different horseshoe bat roost sites, to get a handle on what actually happens with that virus. And even if that particular virus is not zoonotic, because we don't properly understand how it got out of the bats into the human population in the first place, it would be really, really useful to know what it actually does in the bat population, so we have a better idea of how not to end up all locked up again for another 2 years.
Excellent. Yeah, well, I'll get in contact with them then, and we'll sort something out after the meeting. Do you think agricultural colleges and universities, where lots of students have contact with the animals, should do routine testing on the animals now it is not mandatory for students to be tested, so as to keep on top of any changes in the virus in humans and animals?
So we did intensively monitor the students here. So the university ran its own testing program at enormous expense. They spent about 20 million on it. But anyway, so we did monitor the students quite intensively. That's obviously stopped now because the caseloads dropped right off in people, and it's no longer considered so urgent. The species we have that they come into contact with are not ones that are at all susceptible, really. So horses, cattle, sheep, and we do have a variety of small animals. In general, that would be an excellent thing to be able to do, because vet students get all sorts of zoonotic diseases, and we usually think of it as a problem for the vet students rather than it going the other way around, and we probably should think more about it going the other way around, but that's just funding dependent, because all of that, the project I presented to cost £300,000. That's cheap for that kind of work. To do it long term, you're talking an awful lot of money. So we have to make a really good case to do it.
Right, yeah, I can imagine it's expensive. Does the recent spillover of sewage into our rivers cause a concern for transferring the virus?
Yeah, and that was a massive concern, and there's a number, we're not the only ones working on this, there have been a number of projects monitoring sewage levels, and it's been Vincent at UCL who's been doing the river monitoring. For SARS, fortunately, when they monitor the rivers, when they monitor the sewage outflows upstream of the water processing plants, they do pick up SARS. When they monitor it downstream of the water processing plants, they're not picking up anything at all. So the sewage treatment's quite effective at destroying SARS, which is a good thing. But obviously, if you've got storm overflow of the sewage plants being bypassed, like has happened recently and keeps happening, then that system gets bypassed. And it's one of the reasons we included otters, because they are in the water, and mink. And it was one of the big concerns that those aquatic species might be being exposed and might pick it up from exposure to human sewage. What it looks like is the virus loads, even though it's in there, it's very, very diluted and very damaged because it's in big volumes of water. So it's not a high enough infectious dose to be getting into the animals and causing infection by and large. So for SARS, that certainly it's looking like a lower risk, but it is always an issue with sewage overflows into rivers. We end up with noroviruses in shellfish, and then people eat the shellfish and get noroviruses. We then end up with noroviruses in other things that eat the shellfish, like seals and things like that. And we're also quite concerned at present with the avian influenza outbreak in the seabirds, that we're seeing a real uptick in cases in carnivores like otters and foxes. Oh, foxes, I forgot to mention foxes. The APHA have been monitoring all the fox samples for SARS, and fortunately we haven't detected it at all, which is a good thing, because in the UK we've got quite an unusual ecology with our urban foxes. So they were regarded as quite a high risk species for SARS. And we're currently looking with some concern at a similar range of species because of avian influenza. And we see sporadic cases of that, and that's a different disease and a different concern, but the same basic problem in that I obviously feel that we need to get a grip on our wildlife disease monitoring and our animal disease monitoring for all of these reasons, and to not just, because DEFRA, the APHA's remit, and they have some very good people, is focused on livestock for trade purposes. And we have a big hole with our small animals, with our wild animals. And the university's kind of plugged that gap with the COVID urgency grants, but that's not a long-term plan. That actually needs to be built into the system.
Yeah, sure. From your presentation, it is clear that wilder populations of mammals are less susceptible than those mammals that are intensively farmed or kept. Do you believe this is due to wilder animals having less contact with others of the same species, or do you believe that stress in captive and farmed and kept animals has a part to play in reducing the effectiveness of their immune systems?
I think it's population density primarily. It's the number of animals that are in close contact with each other. I think that's by far and away the overwhelming factor here. And I think most people who've been looking at the deer outbreak are leaning towards that as the explanation for why we've seen that outbreak in the US, because their population density is so high. Captive kept animals don't necessarily have poor animal welfare. I mean, I've worked in farming in the past, and the scale of the system is not kind of the thing that makes the difference with welfare. It's the concern of the person who looks after the animal that makes the biggest difference. And worked a lot in zoos, and captive animals don't necessarily have poor welfare. They don't necessarily have increased stress loads. The zoo animals live a hell of a lot longer. They're fatter, they breed better, all the things that you regard as... Vets have a bit of a different... Vets focus on health when they talk about welfare rather than behaviour, which tends to be what quite a lot of the public focuses on when they use the word welfare. So yeah, I really think with the whole captive versus wild thing, it's population density is the overwhelming factor there to be tested, but that's where I think it's going, where it is.
Okay. I think this is the last one on the chat at the moment, unless anybody else has got any questions. Somebody's wondering if caving equipment and clothing is possibly responsible for transferring viruses from bats.
It's certainly possible, and there was an outbreak in between SARS 1 and 2 of something that was probably a sub-ecovirus in China, and it was picked up in people who were going into caves they were mining bat guano, the feces, which is used for fertilizer. So it's certainly a risk going into caving sites and bringing things out on equipment. And we know that the fungal disease, white nose fungal disease in the US is spread that way. Fungi are much better at surviving on surfaces than most viruses are. And we've been talking about SARS and influenza, which are envelope viruses, which are pretty pathetic when it comes to environmental survival. There are other viruses like pox viruses that survive very, very well and for a long time in the environment. So those viruses do get spread on equipment or gear. Not that I know of a bat pox virus. I'm sure there is one, but everyone's been worried about monkeypox recently. But we have cowpox in the UK in our rodents, and it lasts very well in environment and spreads quite effectively on fomites, on things, on clothes, on hands. So cleaning your caving equipment is really important, both for your protection and the protection of the next cave site you go to.
Excellent, thank you very much. Well, that's all the questions on the chat, so I'll hand back over to Jo.
Okay, so it just leaves me to say thank you to you, Rachel. I really appreciate you sharing your knowledge and your expertise. It's been very valuable. I know you've really enhanced my understanding of this topic. I've had questions that I've played with in my head during the night over the last 2 years, so I really appreciate that. I'm just going to ask Vanessa, did you want to say any words? You don't have to, because I know you've written a question. Did you want to say anything before Rachel goes?
Yes, I just wanted to thank Rachel on behalf of both Badger groups for giving up her very valuable time this evening. I found it very enlightening. It's the second time I've heard Rachel. And the one thing that I think she's to be congratulated on is that although she's a very, very clever academic, she's been able to talk to us at a level that anyone could understand. And that is hugely valuable. So thank you, Rachel, very, very much.
Fine. I wish my students were as interested.
Thank you.
Thank you for inviting me. It's a pleasure.